This video is about the examination of freshly collected semen from any of a variety of species, such as bulls, rams, and bucks. We prefer to see the examination and evaluation of semen as different processes with different purposes. Semen examination is the process whereby information about the quality of a semen sample is collected. Semen evaluation, on the other hand, is the process of interpreting the information collected during semen examination and drawing useful conclusions about the semen sample and its donor. Numerous causes, of which we will mention a few, may alter the quality of a semen sample one examines, compared to the quality it would have had if the donor served a female in estrus. Such causes altering semen quality may relate to the process of semen collection or thereafter to the process of semen examination. Such alteration in semen quality would, of course, detract from the quality of conclusions that can be drawn about the semen and its donor. From the moment the semen sample is collected until the examination has been completed, one should strive to minimize the change in semen quality compared to what it would have been the moment the animal served an estrus female. The aim of this video is to demonstrate one way of examining semen that would minimize the discrepancy between the assessed quality and the real quality of the sample at the time of ejaculation. Before examining semen, we should look at a few prerequisites. We will look at the equipment needed for a routine semen examination, and thereafter at a few generic techniques that make the work easier and more reliable. For a routine semen examination, one needs the following equipment: a microscope, preferably with phase contrast on the ×10 or ×20 objective, and on the ×100 oil objective, and a bright field ×4 objective. One also needs a warm stage set to 37 degrees Celsius and a warm pad at approximately 30 to 37 degrees Celsius. On the warm pad there should be a box with 22 by 22 mm cover slips and a box with good quality microscope slides with one end frosted and preferably smoothly cut edges rather than polished edges. The microscope slides and the cover slips should be kept in their boxes, which should be closed immediately after removal of a slide or cover slip. One should have a number of new, clean Pasteur pipettes on the warm pad. It is good practice to keep all unused Pasteur pipettes facing the same way on the warm pad, and once one has used a Pasteur pipette and intends using it again, put it down next to the other pipettes, but with its end pointing in the opposite direction. On the warm pad, also have a suitable extender, such as one containing, among other ingredients, egg yolk, tris, citric acid, sodium citrate, and fructose, and a bottle with carefully prepared eosin nigrosin stain. Finally, one should also have a packet of pH paper strips with a half log unit resolution in the range 5 to 8. Spermatozoa metabolize and function optimally at a temperature of about 37 degrees Celsius. From the time of ejaculation, the semen sample should be kept at the slightly lower temperature of 28 to 30 degrees Celsius. Keeping the semen sample at this lower temperature until it is examined suppresses the metabolism of the spermatozoa and thereby prolongs their lifespan sufficiently to permit accurate examination. Rapid excessive cooling of spermatozoa causes cold shock, which irreparably damages them. Avoid cooling the semen rapidly, especially if the semen is cooled to below 15 degrees Celsius. Cold shock most easily occurs if the concentration of spermatozoa in the ejaculate is very high and the volume thereof low. There are various easy and practical ways of keeping semen at 28 to 30 degrees Celsius. If the general environment is warm, with a temperature in the order of 28 to 30 degrees Celsius, the tube with semen may simply be placed on the bench where the examination takes place. If the environment is somewhat cooler, the tube with semen may be placed in a water jacket at 28 to 30 degrees Celsius, subsequent to which the jacket and tube may be allowed to gradually cool to environmental temperature during examination. A water bath may be contrived from a wide-mouth flask. A piece of styrofoam floating on the warm water inside the wide-mouth flask is useful to suspend the tube with semen in the water. First choice, of course, is a dedicated water bath. Dry warming blocks set to the correct temperature are also useful. Instead of the previous methods, an assistant may gently hold the tube with semen in the hand, taking care not to tightly clamp the tube, which will raise the spermatozoal temperature to 37 degrees Celsius and shorten their lifespan. Apart from the equipment for handling and holding the semen, one should also have a data capture sheet, a pen, a waterproof marking pen with which to mark semen tubes, and a sharp pencil with which to mark smears. Having looked at the required equipment needed to examine semen, let us look at those generic techniques that make one's examination easier and more reliable. Any person wishing to examine semen should know how to properly handle a Pasteur pipette. If one carefully lowers a Pasteur pipette over some fluid until the tip just touches the meniscus, about 15 millimeters of fluid will spontaneously rise into the pipette by capillary action. The fluid will tend to stay there when the pipette is withdrawn from the meniscus, even with the back end of the pipette left open. This spontaneous rise into the pipette is due to the adhesive forces between glass and fluid molecules, and due to cohesive forces among fluid molecules at the meniscus inside the pipette, which tends to keep the meniscus as small as possible. There are two problems with this bit of fluid that rise into the thin part of the pipette by capillary action compared to fluid that extend into the thicker part of the pipette as well. The first problem is that the thin column of fluid is more prone to rapid chilling than fluid in the thicker part of the pipette. The second problem, with this bit of fluid rising into the thin part of the pipette only, is that it is more difficult to get out of the pipette when needed than if the fluid extended into the thicker part of the pipette as well. Preferably, one should draw more fluid into the pipette than would enter by capillary action. Although one could suck fluid into the pipette, this could leave one with an undesired mouthful. Rather not suck on the pipette with your mouth. Although using a suction apparatus, such as a syringe attached to the pipette, works well for the crude handling of the fluid, one sacrifices the fine control that allows one to let precisely the desired small amount of fluid out of the pipette when needed. Let us draw sufficient fluid into a Pasteur pipette to enter the thicker part of the pipette without having to apply suction. The first method is used when the fluid is quite deep in the reservoir tube. Simply lower the tip of the pipette into the fluid and let the fluid spontaneously enter the tube from the bottom to the desired height. Of course, the maximum height the fluid would rise to is about the same as the depth of the fluid in the reservoir. Before withdrawing the pipette from the reservoir, close its back with your finger. The problem with this method is that often there is only a small, and hence shallow volume of fluid in the reservoir. It is still possible to draw fluid into the pipette until the length of the fluid column in the pipette is larger than the height of the fluid in the reservoir. To do this, lower the pipette into the fluid in the reservoir. Then tilt the reservoir until the pipette approaches a horizontal position, which will allow fluid to spontaneously flow into the pipette. As long as a Pasteur pipette with fluid is held horizontally, there is no pressure difference between the menisci, causing the fluid to remain in the pipette, although both ends are open, even if the pipette is almost full of fluid. The rate of efflux from such a tube can be crudely controlled by raising the back open end of the pipette until the meniscus nearer its back end is sufficiently higher than the tip of the pipette. Fluid can be expelled from a Pasteur pipette in a finely controlled way, to the exact amount required, and at the precise moment required, by holding the pipette horizontally in the hand, and pressing one's finger against the back opening of the pipette. Presumably, the hand holding the pipette warms the air above the fluid, while the finger prevents the air from escaping. The warming air expands, and because one's finger prevents the air from escaping, the air applies a force to the large meniscus in the thick part of the pipette, which is converted to a higher pressure onto the fluid in the thinner part of the pipette, pushing the fluid out via the tip of the pipette. If the air can no longer propel the fluid, one lifts one's finger away from the back of the pipette and permits the warm air to equilibrate with cooler air from outside the pipette. By repeating the process, more semen is expelled until the pipette is empty. This fine control over the rate and extent of efflux permits one to use the Pasteur pipette to make a drop of any desired size by carefully expelling the desired tiny volume of fluid from the pipette. Note how the size of the droplet that was pushed out of the tip remains the same while one removes one's finger from the back of the pipette, only to increase again when one applies renewed pressure. It is useful to stick the cover slip onto a microscope slide by means of a very small drop of clear fluid. Reasons for this are to render the cover slip easier to handle and keep warm, prevent it from getting blown away by a breeze, or fall off the slide when the slide with the cover slip is inverted. Whenever one needs to mix a drop of semen with a drop or 5 of something else, such as semen extender or eosin nigrosin, it is easier to mix the droplets on a cover slip stuck to a slide rather than on a microscope slide itself, because drops remain relatively globular on a cover slip but spread out into a thin flat layer of fluid on a microscope slide. It is easier to work with such a globular droplet on a cover slip, but quite impossible with a thinly spread out droplet on a slide, as the thin layer will not spontaneously enter a Pasteur pipette. The Pasteur pipette is useful to transfer fluids onto a cover slip and mix them by repeatedly permitting the fluid to spontaneously enter the horizontally held pipette before expelling the fluid onto the cover slip again. One may use a Pasteur pipette to selectively collect material from the bottom of the fluid inside a tube, for example, to collect debris or pus floccules from the bottom of a tube with semen. To render the effect clearly visible, the tube shows a clear fluid with debris in the bottom. To do this, use a new Pasteur pipette that is dry inside. Firmly close the back of the pipette with a finger while lowering the pipette into the semen until its tip is at the bottom of the tube. During its descent, the tube will remain dry inside except for a little bit of fluid entering by capillary action. Once the tip of the pipette has reached the bottom of the tube, briefly let go with the occluding finger before occluding the back of the pipette once again. During the brief period during which the back of the pipette was open, a little bit of fluid could spontaneously enter the pipette via the tip, drawing debris or pus with it. After having closed the back of the pipette again, withdraw it from the tube. Because the back of the pipette was quickly occluded again, the amount of fluid that entered the pipette would be small enough to prevent excessive dilution of the debris or pus. The techniques just demonstrated are useful in the evaluation of semen, irrespective of the species from which the semen was obtained, or whether the semen has been processed, for example, by having been frozen and thawed. In this module, we will go through the steps of performing a routine examination of a freshly collected semen sample from a bull. At first, we will consider a few pertinent matters pertaining to the process of collecting the semen sample, and then examine the sample without the aid of a microscope. Preferably, the person collecting an ejaculate should also examine it, because observations made while the semen sample is being collected are helpful in interpreting findings during examination. These data include whether the semen was collected by means of transrectal massage, an artificial vagina, an electroejaculator, or a combination of these. The collector should also record whether the glans penis protruded from the sheath at the time of collection, or whether the bull ejaculated into the sheath. The collector should attempt to find out what the interval is since the bull previously ejaculated and record the finding. Finally, the collector should record whether or not some part of the ejaculate spilt during collection, and if it did, what fraction was collected. If different persons have to collect and examine the semen, the collector should record this information on the data capture sheet, which should accompany the semen to the examiner. It is good practice to label the tube containing the semen with the identification of the bull and the time of collection. Examine the sample as soon as possible after collection, and keep it at 28 to 30 degrees Celsius until the examination has been completed. The first phase of the semen examination is done with the naked eye and is called the macroscopic examination. Measure the volume directly against the edge of the calibrated tube. Record the volume. If the volume is too small, draw the semen into a Pasteur pipette by means of a syringe and measure the displacement of the plunger of the syringe. Describe the color of the semen. If the color is subtle, hold the sample against a white or black background. Increasing concentrations of spermatozoa in clear seminal plasma will result in a range of colors ranging from colorless through various shades of gray to white and eventually ivory. Some bulls produce yellowish semen due to pigments in the seminal plasma. Because the pigment occurs in the seminal plasma, the supernatant after centrifugation will be yellow. Urine may also result in a yellowish tinge. Fresh blood will cause a pink to red discoloration, and if the semen is centrifuged, a red ring on the white sperm sediment with clear, colorless seminal plasma. Old blood will cause a brownish pink to brown discoloration of the ejaculate. If hemolysis has occurred, it leaves the seminal plasma pinkish-brown after centrifugation. Pus may cause a yellow or greenish discoloration. Record the color. Estimate the consistency of the semen by tilting the tube. The consistency of seminal plasma free of spermatozoa is as low as that of water, which is, therefore, also the lowest consistency possible. On the other extreme, the consistency of a semen sample with a very high concentration of spermatozoa and a minimum amount of seminal plasma is more viscous than cream, which is described as a thick, creamy consistency. Between watery and thick creamy lie thin milky, milky, thin creamy, and creamy consistencies. If the consistency is solely due to spermatozoa suspended in seminal plasma, the consistency provides an estimate of the sperm concentration. Apart from spermatozoa, viscous material such as pus may also increase the consistency of semen. Record the consistency. Provided that the concentration of spermatozoa in the semen sample is high enough, and at the same time that the percentage motile spermatozoa is high enough, and finally that one's eyesight is good enough, one may see marbling in a semen sample. To look for marbling, one should intently stare at a small area of the semen through the wall of the tube. Such staring may reveal that the semen is not homogeneous, but mottled, reminding one of the appearance of marble. Close inspection may reveal that the irregular appearance moves with time in what appears as a granular or swirling motion. This phenomenon is called marbling. Another way of looking for marbling is to tilt the tube with semen to near horizontal and then tilt it slightly more upright to form a shallow edge of the semen along the wall of the tube. Close inspection of this shallow edge from underneath against background light renders marbling more easily visible. Marbling may be absent if the concentration of spermatozoa is too low or the percentage motile spermatozoa is too low, or both. Record the marbling as absent, slight, or distinct. The degree of marbling serves as a cross-check of mass motility, which is assessed microscopically, and the two should correspond closely. Mucus, pus, and debris are usually more dense than semen and therefore rapidly sediment at the bottom of the tube. Inspect the bottom of the tube after it has been kept upright for a few minutes and record such presence. The presence of such substances should correspond to the findings when a Giemsa-stain smear of the semen is inspected, which will be described later. The normal pH of semen is about 6.8. Therefore, one should use paper with a good resolution in the range 5 to 8. Moisten the indicator areas of the paper with semen and measure the pH to the nearest half-log unit. Record the pH. Carefully smell the semen sample. With experience, one will be able to recognize the normal smell, which we experience as faint. Semen heavily contaminated with debris, such as pus or feces, may smell of the contaminant. We once smelt semen with a strong fetid smell because the concentration of spermatozoa was very high. But all the spermatozoa had been dead and autolyzing inside the epididymal tails of a paralyzed and recumbent ram. One reason for an abnormal smell is urine contamination. If a drop of semen suspected to contain urine is transferred to a microscope slide and warmed with a gas flame, the smell of urine may become more noticeable. Record the smell. This brings us to the end of the macroscopic examination. The data collected so far will contribute directly towards the conclusions about the ejaculate and the donor one will later draw during the evaluation process. The data will also serve as useful cross-checks with data we are about to collect during the remainder of the semen examination. Once finished with a macroscopic examination, one should proceed immediately with a microscopic examination of the semen sample. We will start by examining the motility of the spermatozoa. The first examination to do is that of mass motility. To do this, use the lowest magnification that the microscope is capable of. Using the ×4 bright field objective is ideal. Because the sample is magnified as little as 40 times only, one cannot assess the motility of individual spermatozoa, but rather the collective wave-like effect of large numbers of spermatozoa moving together, reminding one of the seemingly unified motion of a large shoal of fish. Mass motility becomes more distinct the higher the concentration of spermatozoa and the higher the percentage of motile spermatozoa are in the semen sample. Note that sperm tails separated from their heads may be motile, and spermatozoa with aberrant motility, such as spermatozoa with hairpin bent tails, which swim backward, may cause mass movement. To prepare the specimen for the assessment of mass motility, start by removing a slide from the box and placing it onto the warm stage. Stick a cover slip to the slide, near its end, further away from the frosted end, by means of a tiny drop of clear fluid. Place a small drop of semen, having a diameter of about 5 millimeters, on the center of the cover slip. Of course, a drop of watery semen with a diameter of 5 millimeters will be much more flat than a 5 millimeter drop of thick, creamy semen, which will result in a more globose drop. This difference in shape is one of the factors affecting the mass motility. Invert the slide and lower it onto the warm stage in such a way that the drop hangs over one of the holes through the warm stage. Carefully place the warm stage under the microscope and catch it with a table clamp. While gently holding a finger of the left hand against the left near corner of the warm stage to ensure that the somewhat heavy warm stage will move in perfect harmony with the clamps, move the warm stage on the microscope table by means of the table clamp control knobs until the drop is in the optic path. Using the ×4 objective with the light turned low, focus on the surface of the drop that is in contact with the cover slip. Assess the mass motility and score it on unit scores ranging from 1 to 5. If one sees wave movement at all, the score must be 3 or higher. If the waves are slow and feeble, often only seen near the edge of the drop, where the drop is thickest, the score is 3. If the waves are strong and quite rapid, but rounded, and reaching the edge of the drop, the score is 4. A score of 5 is assigned if the waves are very strong and rapid, forming whiplash curls at the edge of the drop. If waves are absent but fairly distinct movement of individual spermatozoa is seen, the score is 2. The score is 1 if indistinct but definite movement of individual spermatozoa is seen. The score is 0 if no movement is seen at all. Record the mass motility. The mass motility should correspond well with the marbling. If it does not, believe the better one of the two to best reflect the semen quality. If the marbling was good, but the mass motility was poor, prepare another hanging drop and assess the mass motility therein, making sure that all glassware is clean and maintained at the correct temperature. If the mass motility is still poor, suspect damage to the ejaculate and collect another ejaculate from the animal and evaluate it. Following the assessment of mass motility, screen the sample for cells other than spermatozoa. Do this by inverting the slide so that the drop lies on top of the cover slip. Then lower a cover slip onto the drop, flattening it between this cover slip and the previous one. Place the warm stage with the slide under the microscope and focus at the layer of semen using phase contrast and the ×20 objective. While moving the warm stage under the microscope, look for cells other than spermatozoa floating around among the spermatozoa. Squamous epithelial cells are easily recognized due to their large size and somewhat angular shape. Leukocytes appear round and slightly larger than the diameter of a sperm head. Spermatogenic cells are round and highly variable in size. Epididymal epithelial cells are elongate with rounded ends. Plant material is sometimes seen. Record the presence of cells other than spermatozoa based on this crude screening test, and remove the warm stage from the microscope. By now, we have examined the mass motility, and we have done a screening test for cells other than spermatozoa that may be in the ejaculate. If the mass motility is unexpectedly poor compared to the expectation derived from the marbling and the consistency and color of the ejaculate, prepare another hanging drop from the ejaculate and repeat the assessment of the mass motility. In the next module, we will use the same glassware and proceed with a microscopic examination by examining the motility of spermatozoa individually. Having completed the first part of the microscopic examination, which is the examination of mass motility and screening the undiluted semen for cells other than spermatozoa, we will proceed immediately to examine the motility of spermatozoa individually. The method demonstrated in this module is systematic and repeatable. While keeping the slide with its associated cover slips on the warm stage, prepare the specimen from which the individual sperm motility will be estimated. Onto the cover slip, underneath which you did a crude search for cells other than spermatozoa, place a few drops of a suitable semen extender that had been pre-warmed to about 37 degrees Celsius. Place the drops into one another to form a single blob. We use Trilodil or Bilodil, both from Minitube, but many others would be suitable. If the extender is not at 37 degrees when placed onto the cover slip, leaving it there for half a minute or so will bring it to 37 degrees Celsius. Once satisfied that the temperature of the extender should have reached 37 degrees Celsius, mix the semen before drawing some into a Pasteur pipette. Place a suitably small drop of semen into the extender. The volume ratio between extender and semen cannot be prescribed, as it depends on the concentration of spermatozoa in the ejaculate. If eventually the specimen under the microscope is too densely populated with spermatozoa to enable one to assess the motility of individual spermatozoa, one should prepare another specimen, this time using relatively more extender and relatively less semen. Mix the fluid by spontaneously filling a Pasteur pipette and evacuating its contents repeatedly. Before preparing a specimen for the examination of individual sperm motility, we will explain the process diagrammatically. Here is a microscope slide seen from the front, with the first cover slip stuck to it by means of a bit of clear fluid. Here is the second cover slip that flattened the drop of semen used to assess mass motility in order to create a layer of semen that we screened for cells other than spermatozoa. On top of the second cover slip is the globule of extender mixed with semen. Place a new cover slip onto the microscope slide next to the other two cover slips. Place a small drop of about 3 millimeters in diameter onto the center of the new cover slip. Lift the cover slip and invert it. Carefully lower the cover slip while holding it parallel to the slide until the drop of semen in its center touches the slide. Let go of the cover slip to flatten the droplet of semen. In this way, the drop of semen should cover all of the undersurface of the cover slip without trapping air bubbles and without having semen flowing from underneath the cover slip. Let us take another look at the schematic drawing of the slide with the earlier cover slips and the cover slip under which the drop of extended semen is flattened, and view the latter cover slip from the top. One will understand that once one lets go of the cover slip, there will be a radial current from the position of the droplet towards all aspects of the periphery of the cover slip, carrying immotile and some aberrantly motile spermatozoa peripherally in the process. This current would therefore result in a higher proportion of spermatozoa being immotile near the periphery than under the centre of the cover slip, which one should take into account when assessing the motility. If the drop was too large and semen escapes from underneath the cover slip, a disproportionately high number of immotile or aberrantly motile spermatozoa will be carried from underneath the cover slip, creating the risk of an over-optimistic estimation of the percentage of progressively motile spermatozoa under the cover slip. Preparing the semen for the examination of individual motility in this way will avoid air bubbles being trapped under the cover slip. Such air bubbles will make examination difficult, and they may affect the motility. Now that we have seen a diagrammatic exposition of how to prepare the sample for individual motility, let us see the real actions. Although one should include in one's assessment the spermatozoa that immediately drifted to the edge of the cover slip upon flattening the droplet, there will, over time, be an increasing and spurious accumulation of immotile spermatozoa at the edge. This is caused by progressively motile spermatozoa that happen to swim towards the periphery of the cover slip to simply continue doing so without turning around once near the edge. This way, these peripherally directed progressively motile spermatozoa become trapped among already immotile spermatozoa, and also become immotile, thereby causing a progressively widening zone of immotile spermatozoa. To avoid this artifact, start evaluating the motility by first looking at the edge of the cover slip and then assess a number of fields along the west edge to center radius or along the east edge to center radius, as these radiuses are mechanically easy to follow under the microscope. Ideally, the preparation for the examination of the motility of individual spermatozoa should have a thin monolayer of spermatozoa, all in focus at the same time, and with about a sperm's length between neighbouring spermatozoa, as seen here. Having the spermatozoa too densely populated impedes the ability to estimate the percentages of progressively motile or aberrantly motile spermatozoa. Having the spermatozoa too sparsely populated may cause excessive variation in motility from one field to another. Adjust the microscope to phase contrast with a ×20 objective in the optic path. Having selected the west edge to center radius or the east edge to center radius, select a number of microscopic fields along that radius and estimate the motility of each selected field. Perhaps best would be to use each of the 10 or 11 neighbouring fields from the edge to the centre of the cover slip. This may, however, be quite time-consuming, and, as an alternative, every second or every third field along the radius may be selected. Imagine a semen sample in which exactly all spermatozoa are progressively motile. Of course, the percentage progressively motile spermatozoa will be 100, and the ratio between the number of progressively motile spermatozoa and the number of spermatozoa that are anything but progressively motile will strive to infinity against 0. Now imagine a semen sample in which 99% of spermatozoa are progressively motile and 1%, that are anything but progressively motile. The ratio between the number of progressively motile spermatozoa and the rest will be 99 against 1. In the same way, for a sample with 95% progressively motile spermatozoa, the ratio between the number of progressively motile spermatozoa and the rest is 19 against 1. Similarly, the ratio will be 9 against 1 for 90%, about 6 against 1 for a sample with 85% progressively motile spermatozoa, 4 against 1 for a sample with 80% progressively motile spermatozoa, then 3 against 1 for 75%, 2.5 against 1 for 70%, about 2 against 1 for 65%, 1.5 against 1 for 60%, an equal ratio for 50%, and something between 1.5 against 1 and an equal ratio for a sample with 55% progressively motile spermatozoa. In a similar way, it is easy to complete the table of ratios for samples with 45%, 40%, 35%, 30%, 25%, 20%, 15%, 10%, 5%, and 1% progressively motile spermatozoa. We find it easier to estimate the ratio rather than directly estimating the percentage, and then to convert the ratio to a percentage. In this particular field, the ratio between the number of progressively motile spermatozoa and those that are not progressively motile is about 3 against 1, which converts to 75% progressive. Record this percentage. Having estimated and recorded the percentage of progressively motile spermatozoa in the field, estimate the ratio between the number of aberrantly motile spermatozoa and all other spermatozoa combined, irrespective of whether they are progressively motile or immotile. In this field, this ratio is about 1 against 99. Convert the ratio to a percentage, which is 1% in this case. Record the percentage next to the record of the estimated percentage of progressively motile spermatozoa. Move the warm stage until the next selected field, closer to the center of the cover slip, is in the optic path. In the same way as used for the previous field, estimate and record the percentages progressively motile spermatozoa and aberrantly motile spermatozoa in this field. In a similar way, estimate and record the motility of all selected fields along the radius. Here we see the percentages of progressively motile spermatozoa and the percentages aberrantly motile spermatozoa of each of 10 fields that were evaluated. Then calculate and record the average percentages of progressively motile spermatozoa and aberrantly motile spermatozoa. On average, this sample has about 47% progressively motile spermatozoa and about 4% aberrantly motile spermatozoa. Subtract the sum of these two averages from 100 to calculate the average percentage immotile spermatozoa and record the percentage. Once one has studied and mastered this method, one may, depending on the need for the examination, simplify the method. For example, by not evaluating each microscope field along the equatorial radius, but only every second field, which we feel will be less likely to yield a biased result than merely evaluating any few fields or only the central field. Having completed the examination of the motility of spermatozoa individually, cross-check the results against the related tests done earlier. If the individual motility is poor compared to the expectation derived from the mass motility and marbling, make another preparation for individual sperm motility from the ejaculate and repeat the examination. Immediately after the assessment of the motility of individual spermatozoa, while the ejaculate is still fresh and at hand, we will proceed to make a smear for the microscopic examination of sperm morphology. We start by temporarily removing the slide that we have hitherto been using from the working surface of the warm stage and place it elsewhere on the warm stage to keep warm. Now transfer a new microscope slide from the box to the working surface of the warm stage, placing the slide underneath the one that we have hitherto been using. We will refer to the new slide as the slide and the one with the cover slips as the spreader. We will refer to the frosted end of the slide or spreader as the back end and the opposite end as the front end, whereas we will refer to the long edge of the slide nearer the operator as the near edge and the one further away as the far edge. As soon as the slide has been placed on the working surface of the warm stage, place the spreader on top of it. We will also describe and demonstrate the technique for a right-handed person. Left-handed persons should use the opposite hands and directions as those we do. Transfer a few drops of eosin nigrosin stain onto the cover slip covering the spermatozoa, of which we have earlier assessed the individual motility, forming a single globule of eosin nigrosin. We find it easier to use a new, clean, sperm-free Pasteur pipette to extract the eosin nigrosin from the bottle than trying to use the dropper spout to do so. If one uses a Pasteur pipette for this transfer, one should ensure that one does not contaminate the bottle with eosin nigrosin with spermatozoa, as such spermatozoa may later on be inadvertently transferred to the smear of another male semen. Stir the semen sample before transferring a small drop thereof to the globule of eosin nigrosin by means of a warm Pasteur pipette. Use the Pasteur pipette that was used to transfer the eosin nigrosin to the cover slip to mix the globule containing eosin nigrosin and spermatozoa while the globule is being incubated on the warm stage at 37 degrees Celsius for about 2 minutes. During these 2 minutes, spermatozoa that have selectively permeable membranes and are presumably alive will prove their status by being able to keep the large eosin molecules out of the spermatozoa, thereby permitting the spermatozoa to retain their natural white color. During these 2 minutes, those spermatozoa of which the cell membranes have lost their selective permeability and are presumably dead will prove their status by not being able to keep the eosin stain out and hence lose their natural white color and stain red. After the 2 minutes have lapsed, permit the globule to be spontaneously drawn into the pipette. Lift the spreader from the slide and put it down on the warm stage away from the working surface. Transfer a drop of the eosin nigrosin sperm mixture onto the slide near its frosted end. The drop must be of a volume that will spread on the slide to cover an area with a diameter of about 6 millimeters. Hold the front end of the slide at its tip with the tips of the thumb and index finger of the left hand in such a way that the far edge of the slide is near the far edge of the warm stage and the back edge of the slide flush with the back edge of the warm stage. Pick up the spreader with the right hand, holding it nearer its back than its front. Hold the spreader with the pad of the thumb on the near edge and the pads of the index finger and middle finger on the far edge. Place the front edge of the spreader on the slide in front of the droplet. While holding the edge of the spreader down onto the slide, with the back of the spreader tilted until the angle between spreader and slide is about 45 degrees, Pull the spreader backwards until its front edge is about 1 millimeter away from the front of the droplet. While holding the front edge of the spreader down onto the slide, tilt the spreader more horizontally until the whole droplet has spread sideways along the back of the spreader. Gently sweep the spreader along the length of the slide from where the droplet was to the front end of the slide. While sweeping, gradually raise the back end of the spreader until the angle between slide and spreader has reached about 45 to 60 degrees by the time the front edge of the spreader reaches that of the slide. During the sweep, one must take care not to let the spreader lose contact with the slide. After the sweep, lift the spreader from the slide and put it elsewhere on the warm stage, in case the smear turns out to be unsuitable and you need to use the back edge of the spreader to make another smear. While leaving the smear on the warm stage to dry at 37 degrees Celsius, inspect it from above. It should be clear that the whole droplet was spread sideways along the edge of the spreader and thereby contributed to the smear. The smear should have a distinct, fairly dark blue color, as a dark background makes it easier to see the spermatozoa. The smear should be thinner nearer the back of the slide, becoming progressively thicker towards the front. It is quite normal to have some backflow from the front edge of the slide. Once the smear has dried, put it under the microscope while keeping it on the warm stage to permit further drying at 37 degrees Celsius. Use the ×20 objective and either bright field or phase contrast. Focus the microscope. Assess the smear and ensure that, for the most part, it has no scratch marks or other signs of mechanical damage to spermatozoa, such as bent tails with all the spermatozoa lying parallel to one another. Assess the spread of spermatozoa on the smear and confirm that there is a suitable area. Having made the smear so that it is thinner near its back end and thicker towards its front end, increases the probability of finding areas that are neither too thin nor too thick somewhere on the smear. In the area with a suitable spread, the spermatozoa should be randomly directed, and there should be about a sperm's length between neighboring spermatozoa. Once one has confirmed that the smear is sufficiently well made to evaluate, identify the smear by writing the identification number or name of the animal and the date on the frosted end. If more than one ejaculate is collected from the same animal on the same day, also write the number of the ejaculate on the slide. Allow the smear to dry further and thoroughly by leaving the smear in a dry, dust-free, well-ventilated area with no wind for a few hours. It is a good idea to leave it standing on a piece of absorbing paper in an oblique vertical position, with the surface on which the smear was made towards the bottom. If conditions are not suitable for this method, smears can be supported vertically in a slit cut into a sheet of styrofoam and kept that way until needed. Once the eosin nigrosin smear is secured in a dust-free, windless place to dry completely, make a smear that will later be examined for the presence of cells other than spermatozoa. Having made the eosin nigrosin smear, one needs to prepare a smear that will be used to determine whether or not there are cells other than spermatozoa in the ejaculate, and, if they are, what types they are. The first question to ponder is, from where in the ejaculate one should collect the alicot from which one will make the smear. 3 possible sites of collection that each has its value, depending on the information one wishes to derive from the smear, may be used. Firstly, one may collect an alicot from anywhere in the thoroughly mixed ejaculate. A smear made from such an alicot will provide an overall average, unbiased impression of the occurrence of cells other than spermatozoa in the ejaculate. With some ejaculates, one may decide to collect an aliquot from the bottom of the tube with semen after the tube has been upright for a few minutes, during which debris, pus floccules, and so on, had the opportunity to sink to the bottom of the tube. A smear made from such an aliquot will provide a biased impression about cells other than spermatozoa in the ejaculate. It would better answer the question whether or not there are cells other than spermatozoa lurking somewhere in the ejaculate than it would answer the question what the overall average status of the ejaculate is with respect to cells other than spermatozoa. Especially in watery samples, one may decide to collect the aliquot from which to make the smear from the bottom of the tube with semen after it has been centrifuged. Clearly, such a smear made from the sediment would not at all provide a fair reflection of the prevalence of cells other than spermatozoa in the ejaculate, except, of course, if there is no sediment at all, or if the sediment contains no cells other than spermatozoa, in which case we may fairly conclude that there are no cells other than spermatozoa in the ejaculate. What the sediment smear will provide us with is a highly sensitive answer on the question whether or not there are any cells other than spermatozoa anywhere in the ejaculate. For the purpose of the current demonstration, we will assume that routine prevails, and that the aliquot is therefore withdrawn from the center of a well-mixed ejaculate. There exist various ways of making a smear that is suitable for assessing the status of the ejaculate with respect to cells other than spermatozoa. These methods have in common that they are made from undiluted semen, and that they provide a smear with thinner and thicker areas on it. The method described now is one of many that work well. Remove 2 new slides from the box, one of which will serve as a slide on which to make the smear, and the other as a spreader. It is easier to make the smear while holding the slide in one's hand in the air. It is not necessary to keep the slide on the warm stage, as the smear will only be used to assess cells other than spermatozoa, and these cells are not affected if the slide is cool. Transfer a small streak of about 10 millimeters long and 2 millimeters wide of semen from the pipette to the center of a new clean slide. Place the lower surface of a clean spreader onto the slide, in such a way that the spreader lies at a right angle across the slide, flattening about 1/3 of the streak of semen between slide and spreader. Then sweep the spreader towards the frosted end of the slide with a turning motion that would slide the spreader off the surface of the slide without covering the frosted end of the slide. This part of the slide usually provides a spread of cells that is optimal for the identification of the type of cells other than spermatozoa. Now place the edge of the spreader on the slide to the front of the remaining part of the streak of semen. Turn the spreader sideways so that its edge on the slide lies at an angle of about 45 degrees relative to the long axis of the spreader. The purpose of this rotation is to move the corners of the spreading edge away from the long edges of the slide, so that the edges of the forthcoming part of the smear are away from the long edges of the slide, which will facilitate evaluation of all parts of the smear, including its edges. Tilt the spreader until the angle between the slide and the spreader is about 45 degrees. Pull the spreader back, parallel to the long axis of the slide, until the spreader has spread the remaining part of the streak of semen behind its edge. Move the spreader forward, parallel to the long axis of the slide, for about 7 millimeters. Lift the spreader and put it down about 5 millimeters nearer the front edge of the slide. Again move the spreader parallel to the long axis of the slide for about 7 millimeters before lifting it and putting it down again 5 millimeters closer to the far end of the slide. Repeat the previous part of the smearing process until the far end of the slide is reached. This way of making the smear to the front of the position of the streak will yield a number of thick lines that progressively become shorter, narrower and thinner. Each of these thick streaks will be neighboured by a thinner zone. Inspecting the thicker areas provides a sensitive indication of whether or not there are cells other than spermatozoa in the ejaculate, whereas the thinner areas permit one to identify the types of cells. Use a pencil to mark the smear with the identification of the bull and the date. If more than one ejaculate was collected on the same day, also write the ejaculate number on the slide. If the smear was made from anything other than a truly random aliquot from the ejaculate, for example, from the spontaneously formed sediment or from the sediment after centrifugation, the information should also be written on the slide. Dry the smear thoroughly by leaving the smear in a dry, dust-free and well-ventilated area with no wind for a few hours. If the smear is left to dry spontaneously, it is a good idea to leave it standing on a piece of absorbing paper in an oblique vertical position, with the surface on which the smear was made towards the bottom. One may also dry the smear by blowing against the undersurface of the slide with warm air from a blow dryer, or gently warming the undersurface of the slide with a gas flame. Once dry, place the smear on a horizontal rack and cover it with a layer of the fixative for one of the rapid Giemsa-like stains for about 10 minutes. Semen is notoriously prone to wash off the slide if one attempts fixing the smear before it has dried completely, or if one fixes it for too short a period of time. After 10 minutes, hold the slide vertically to let the excess fixative flow off the front edge of the slide. Return the slide to its horizontal position. Cover it with a layer of red stain from the set of rapid Giemsa-like stain. After about 20 seconds, let the excess stain run off the front edge of the slide by holding the slide vertically. Once again, return the slide to a horizontal position. Cover it with a layer of blue stain from the Giemsa-like set. After about 20 seconds, hold the slide vertically under cold running water to wash the stain off the smear. Dry the smear by letting it stand obliquely upright, with the smear facing down. The smear may also be dried by means of a blow dryer. Once certain that the eosin nigrosin smear and the smear stained with a Giemsa-like stain have dried completely, lay them on a horizontal surface and mount part of each under a cover slip, using a suitable mounting medium made from mounting microscopic specimens. Remove a large cover slip from its box. Open the bottle containing the mounting medium and, by means of a suitable rod, transfer a thin streak of mounting medium to the equator of the cover slip. Invert the cover slip over the smear. Once the streak of mounting medium touches the slide, let go of the cover slip and allow the mounting medium to disperse spontaneously. Similarly, mount the inscription to render the identification permanent. Mount the smear stained with a Giemsa-like stain in the same way as the eosin nigrosin smear. Do not use too much mounting medium, as that would escape from underneath the edge of the cover slip, which may impede the microscope objective from reaching near the edge of the cover slip. And if incompletely dried mounting medium accidentally lands on microscope lenses, it may damage them. With all the smears made and mounted, they can be stored in a secure way until it is convenient to evaluate them. At a convenient time, after the mounting medium on the smears have dried, which takes at least an hour, one may perform the microscopic examination of the smears. We prefer examining the eosin nigrosin smear first. Record the identification marks of the slide on the data capture sheet. Record the stain that was used. Start by examining the smear at moderate magnification by using the ×20 objective, using either bright field or phase contrast. Avoid areas of the smear with visible artifacts such as scratch marks or spermatozoa all lying parallel to one another. Once one has identified an area in which the spermatozoa are randomly directed with about a sperm's length between spermatozoa, switch to the ×100 oil immersion objective, once again using either bright field or phase contrast. The sheet on which the sperm morphology data are captured has a zone for the capture of defects affecting the sperm nucleus, another zone for the capture of defects affecting the acrosome or the tail, and a zone for the capture of data pertaining to morphologically normal spermatozoa. Assuming that the operator is right-handed, keep a counter of the type used to count livestock in your left hand. Simultaneously, use the left hand to operate the focus. Use the right hand to operate the table clamp knobs. Also use the right hand to record the findings on the data capture sheet. Click the counter once as soon as a spermatozoon has been examined and its status recorded. Record the morphology of 100 neighboring spermatozoa. With an Eosin nigrosin smear, one may simultaneously record the sperm morphology as well as whether a spermatozoon is alive or dead. Record red spermatozoa with a vertical stripe behind the appropriate morphological defect and a white spermatozoon with a circle. This spermatozoon is white and morphologically normal. For such spermatozoa, one does not have to record anything on the data capture sheet. All one has to do is to press the counter once every time such a spermatozoon is encountered. Although morphologically normal, this spermatozoon stained red. Record the spermatozoon with a stripe behind the row titled Percentage Morphologically Normal Spermatozoa. Click the counter once. This spermatozoon is white and has a piriform head. Make a circle in the row named Piriform. Press the counter once. This spermatozoon is white and has a proximal cytoplasmic droplet. Make a circle in the row named Proximal Cytoplasmic Droplet. Press the counter once. This spermatozoon, stained red, and it has two defects, namely a piriform head and the nuclear crater defect. Record the two defects of this spermatozoon with a stripe behind the row titled Piriform Head and another stripe behind the row titled Nuclear Crater. Because this one spermatozoon has two defects, which both affect the nucleus, make a single stripe in the margin to the left of defects 1 to 12 in order to indicate that there is one nuclear defect in excess of the number of spermatozoa affected by them. Note that one has pressed the counter once only, because only one sperm was examined. This spermatozoon is white, and it has a knobbed acrosome, as well as a proximal cytoplasmic droplet and a coiled principal piece. Make a circle in the row named Knobbed Acrosome, another circle in the row named Proximal Cytoplasmic Droplet, and yet another circle in the row named Coiled Principal Piece. Here we have one spermatozoon affected by three defects, which all happen to affect the acrosome or tail. In order to indicate that we have recorded two more defects than the number of spermatozoa examined, make two circles in the margin to the left of rows 13 to 28, which are the defects affecting the acrosome or tail. Press the counter once only, because only one spermatozoon was evaluated. This spermatozoon, stained red, and it has two defects, namely a piriform head and a stump tail. Record the spermatozoon with a stripe behind the row titled Piriform Head and another stripe behind the row titled Stump Tail. This sperm has a defect of the nucleus as well as another of the tail, and both were marked on the data capture sheet. The fact that the marks refer to only one spermatozoon must be captured on the record sheet. Do this by making a stripe in the shaded row between defects 12, which is the last nuclear defect on the list, and defect 13, which is the first of the defects affecting the acrosome or tail. Click the counter once. This spermatozoon stained red, and it has two types of defects affecting the nucleus, namely a piriform head and nuclear craters. The spermatozoon also has two defects of the acrosome or tail, namely a swollen acrosome with a ruffled edge and a broken tail. Record the spermatozoon with a stripe behind the row titled Piriform Head and another stripe behind the row titled Nuclear Crater. Mark the fact that one red sperm has two nuclear defects with one stripe in the block to the left of defects 1 to 12, to record an excess of one nuclear defect relative to the number of spermatozoa containing them. Record the swollen acrosome with a stripe in the row titled Altered Acrosomes, and record the broken tail in the row titled Fractured Flagellum. Mark the excess with a stripe in the block to the left of defects 13 to 28. Because one or more defect of the nucleus and one or more defect of the acrosome or tail were recorded for this sperm, make a stripe in the shaded row between defect 12, which is the row for the last nuclear defect, and row 13, which is the row for the first defect of the acrosome or tail. Press the counter once to record that one sperm was evaluated. Assuming that we have pressed the counter 100 times and that all the red or abnormal spermatozoa among the 100 that were examined are drawn on the whiteboard and have been marked on the data capture sheet, we may calculate the prevalences of various aspects of sperm morphology for the smear. We do this by recording the number of marks, irrespective of whether they are circles or stripes, behind each of the 28 defects in the right-hand column of the data capture sheet. Now derive the percentage of spermatozoa affected by nuclear defects. To do this, first record the number of nuclear defects present in the 100 spermatozoa that were examined by adding the totals behind each type of nuclear defect. In this case, there were 6 nuclear defects among the 100 spermatozoa examined. Next, determine the total number of circles or stripes made behind nuclear defects in excess of the number of spermatozoa affected by these defects. This is simply the total number of circles or stripes made in the block to the left of all the nuclear defects, which is 2. Subtract the excess of 2 from the total of 6 nuclear defects per 100 spermatozoa and record that there were 4% of spermatozoa affected by nuclear defects. Next, derive the percentage of spermatozoa affected by defects of the acrosome or tail. To do this, first determine the number of defects of the acrosome or tail present in the 100 spermatozoa that were examined by adding the totals behind each type of defect of the acrosome or tail. In this case, there were 7 defects of the acrosome or tail among the 100 spermatozoa examined. Nuclear defects. Then record the total number of circles or stripes made behind defects of the acrosome or tail in excess of the number of spermatozoa affected by these defects. This is simply the total number of circles or stripes made in the block to the left of all the defects of the acrosome or tail, which is 3. Subtract the excess of 3 from the total of 7 defects of the acrosome or tail per 100 spermatozoa and record that there were 4% of spermatozoa affected by defects of the acrosome or tail. Before we can derive the percentage of morphologically normal spermatozoa, we need to determine the number of spermatozoa affected by nuclear defects as well as defects of the acrosome or tail. Do this by recording the total of the number of circles or stripes in the shaded row between defects 12 and 13, which is 2 in this case. Now derive the percentage of morphologically normal spermatozoa as follows. Subtract from the 100 spermatozoa that were examined the percentage of abnormal spermatozoa that were among them. The percentage abnormal spermatozoa is derived by calculating the sum of the percentage spermatozoa affected by defects of the nucleus and the percentage spermatozoa affected by defects of the acrosome or tail, which is 8 in this case. Subtract from the figure of 8 the number of spermatozoa with defects of the nucleus as well as defects of the acrosome or tail, which is 2 in this case. In this way, we derive that there are 94% morphologically normal spermatozoa on the smear. To derive the percentage normal viable spermatozoa, first calculate the number of morphologically normal dead spermatozoa among the 100 that were examined by obtaining the sum of the total number of stripes made in the row titled Percentage Morphologically Normal Spermatozoa, which is 1 in this case. Subtract this total from the percentage of morphologically normal spermatozoa. In the current example, the percentage normal viable spermatozoa is 93. To derive the percentage viable spermatozoa, add to the 93% normal viable spermatozoa the number of circles made behind any of the defects numbered 1 to 28, which is 5 in this case. We need to account for the number of circles recorded in excess of the number of viable but abnormal spermatozoa. This excess is the total number of circles made in the block to the left of defects 1 to 12, and in the block between defects 12 and 13, and in the shaded block to the left of defects 13 to 28. The excess is 2 in this case. Subtract this excess of 2 to derive that 96% of the spermatozoa on the smear were viable when the smear was made. Remove the eosin-nigrosin smear from the microscope. We now shift our focus to the smear stained with a rapid Giemsa-like stain. Indicate on the form whether the smear was made from a drop of the whole ejaculate or from a sediment. Using bright field and the ×20 objective, scan the smear to identify the thick zones. Examine these thick zones in order to determine whether or not there are cells other than spermatozoa on the smear. In this case, there clearly are such cells. If cells other than spermatozoa are present in the thick areas, explore the thinner areas in order to determine whether or not such cells are also present where the smear is thinner. If they are, use the ×100 objective with immersion oil to examine these cells under high magnification. If cells other than spermatozoa were seen in the thicker areas but not in the thinner areas, or the other way around, examine those cells wherever they were found. Record the cell types that are present, as well as a subjective indication about the numbers. We have now finished examining the ejaculate. The methods shown in this video are simple, systematic, and repeatable, and if they are all performed, provide the information needed for an integrated evaluation of the semen quality. This video certainly does not represent the only way of performing a valid or high-quality routine examination of an ejaculate. There exist many more specialized examinations that require more advanced techniques, and they fall beyond the scope of the aim of this video. Once the examination has been completed, one may reflect on the meaning of the results and interpret them and formulate one's conclusions. This process of deriving meaning from the results may be referred to as semen evaluation, which requires sufficient knowledge and understanding of the andrology of the species. Semen evaluation falls beyond the aim of this video. The person doing the semen evaluation may attempt answering a number of questions, such as whether or not all of the results of the semen examination are reliable, whether or not the semen sample that was examined is likely to reflect the best quality semen the animal was capable of providing at the time, and whether or not the findings satisfy the set of criteria that need to be applied for the particular case at hand. If the evaluation reveals that the semen quality is inadequate, the data capture sheet, if properly populated with carefully collected information, will place the examiner in a position where he or she may be able to draw sound conclusions about possible causes of the deficit, as well as the prognosis for improvement, and suggest appropriate advice.